Now we will see the life cycle of the retroviruses. We have already studied the properties and salient features which viruses and which retroviruses which make the retroviruses different from the other viruses, and then its different characteristics. Now its life cycle. With the help of the molecular biology tools, nowadays in the 21st century, the molecular biology tools help us that they have distinctly, we have divided the two phases of the retrovirus's life cycle. First phase, which is consists of the attachment of the virus with the host receptors and then entry inside the body. The other phase is the actual replication process. The virus is distinctly divided into two phases as far as its replication cycle is concerned, or you can say the life cycle is concerned. The first step, the early steps, involves from the cell surface towards the nucleus of the cell. Then the second step, the things which are happening around inside the nucleus. Retroviral particles must bind specifically to their target cells, then cross the plasma membrane. The retroviruses has to cross two different kinds of the membranes, unlike the other viruses which only have to cross the cell membrane, but viruses also have to cross the nuclear membrane. Then the must create their own RNA genome and uncode the core proteins, find their way towards the nuclear membrane, as we already studied that they have also passed the nuclear membrane and then penetrate inside the nucleus. After the penetrating inside the nucleus, of course, they will have to replicate and they will take all the charge of the host replicating machinery for their own purposes. Then the later stage, which is involved, of course, the transcription, transduction, translation, all the other processes which are involved in the normal replication process. After the transcription and transduction, the viral genome has to need to assemble inside the capsid at the one place. After the assembly, the viral genome need to maturation, and after the maturation, there is a process of the budding, and after budding, the viral genome need to release from the cell. But in this case, the retroviruses, they will just integrate the genome of the virus will be integrated with the host. We will see in the diagram next. These are two different stages which are important from the retrovirus life cycle, the early stage and the late stage. First of all, the early stage, the budding, and then the fusion of the virions or viral particles which need to enter inside the nucleus. The viral receptors are there, of course, which are in the host. And then here there are some special kind of proteins, LV1, RE, CEM, FV2 and FV1. Some of the functions of their functions are still unknown, but they are playing their part. They are playing their part, very important part inside. And then uncoating and reverse transcriptation. The nuclear import, of course, after that there is a nuclear import and the virus will be entered inside the second membrane, which is the nuclear membrane. Inside, after entering the nuclear membrane, the virus will just take the charge of the host machinery and then here at this moment, the virus is called as provirus. Then the virus is released and then the transcription occurs. And after the transcription, the transduction and the viral materials, the immature virus is released, which need to be assembled inside, and then maturation and budding happens. And again, all this process will be done inside the cytoplasm because it will be released from the nucleus and then maturation of the virus and release from the cell.